Brilliant. Okay, so I'm going to talk about ion mobility now, and I'm going to start off with a few caveats around here. You're going to see that there's a number of different flavors of ion mobility, and I've only been largely practicing one flavor of that ion mobility sort of during my time, and it's been relatively recent sort of incursion into the ion mobility field. So what I would encourage you to do is there's a number of different reviews that I've listed as I've gone along the way. So I'd encourage people to read around some of those reviews, but also we've heard about ion mobility in several talks already. So, so particularly have a look at those talks. Sven gave a very nice introduction to TIMS as a form of ion mobility. So I'd recommend that. So there's a few caveats sort of right at the start. So in terms of my talk overview, I thought I'd start off by telling you what ion mobility is and why we might need it. One of the key messages is it's another mode of separation, and in particular, it's an orthogonal mode of separation compared to the high resolution mass spectrometry that we might be performing, and also the chromatography on the front end. And one of the things I'll try and get across is just how complementary the three different techniques are together. And when you're doing ion mobility, we can measure a parameter of the ion called a CCS value, stands for collision cross-section, and this tells us something about the structure of those ions in terms of their drift time as they go through a drift tube, but also tells us about, it helps us in terms of the annotation of those ions, and it's another bit that we have in terms of the jigsaw for structural identification. Probably sort of more useful is to show you some of the applications of ion mobility in action, as it were. So how can ion mobility improve lipid separation and identification? And the nice thing is that in terms of lipidomics, there's a number of very nice applications of where people have used ion mobility to improve separation. So I'll start off by looking at separation of complex mixtures. We'll look at some standards, first of all, to see sort of how we can get separation of those standards, and then move on to tissue extracts and blood plasma samples, and show you how we can begin to separate out isomers. So at the moment, what I would say is that a lot of the applications are separating out structural isomers, but there's some evidence that we're actually going to be able to do better than that in the future and be able to sort of separate out isomers according to where double bonds are present, and maybe even stereoisomers as well in the future, and how we can separate out those structural similar compounds. I've got a few slides on what I've described as the next generation of ion mobility approaches, and it's a bit fuzzy where we go from current ion mobility and next generation ion mobility. We've already heard from one of the talks this morning about the cyclic ion mobility, but this type of approach is going to give us a huge improvement in terms of resolution of ions as part of this approach. And I'll also talk about another approach called SLIMS, which is improving ion mobility and making it more available in terms of access to it. And then finally, I'll finish off on just a slide on ion mobility databases. So as I've already mentioned, we can measure CCS values of our ions as they go through the drift tube, and we can use that for structural information. And one of the surprises for me, sort of coming relatively new into this field, is how highly reproducible the in silico determination of CCS values are for structural identification for many compounds. So I'll start off with what is ion mobility spectrometry, or IMS. So it's a gas phase technique allowing the separation of ions based on their mobility through an inert gas under the influence of an electric field. So we saw some examples of this morning about the difference between a piece of paper falling down through the air and a crumpled up ball. I have a slightly different analogy of relevant to anybody in the UK at the moment. We've just reopened our pubs, at least for going outside in England. And if you need to get to the bar, often it's handy if you're a smaller person like myself, you can nip in through gaps and you can get closer to the bar more quickly. Whereas if you're a larger person, you're going to be impeded by the other people that are present there. So with ion mobility, we're not going to use people to impede our ions. We're going to send the ions down a drift tube at least in one of the designs for measuring ion mobility. And we'll accelerate those ions using an electric field. Now, at the same time, the progress of these ions are impeded by a drift gas going in the opposite direction. And what we find is that smaller ions will go through the drift tube more quickly, and the larger ions will be impeded by that inert gas as they go through. It's relatively cheap to make these ion mobility tubes. And as a result, it doesn't necessarily cost that much more in terms of putting it on the front end of our mass spectrometer. So it gives us that added bit of separation for not very much more in terms of cost, and at least in terms of the electronics. I should have said that actually this type of design in terms of ion mobility has been around since the 1950s. So it was Earl McDaniel working at Georgia Tech that came up with some of the original ideas around ion mobility. So it has been around for 60, 70 years, but I think, you know, it's sort of enjoyed a bit of a renaissance recently as we've got better designs sort of coming onto the market. I've already mentioned it's relatively easy to hyphenate with mass spectrometry. You've probably all met ion mobility spectrometry. Last time, you have to cast your minds back now over a year to last time that you were at an airport. So a lot of the explosive detectors are based around ion mobility based approaches because they're cheaper than mass spectrometry in terms of designing them. Last point to make is that separation is typically on a millisecond scale. So you'll see that a lot of the drift times I'll be referring to are sort of 40, 50 milliseconds in total length. So what this means is that we can actually readily hyphenate this with chromatography, where typically we might be separating peaks in terms of the seconds or tens of seconds, and mass spectrometry, where we might be hyphenating things in terms of tens of microseconds, depending on which flavor of mass spectrometry that we're using. So hopefully that gives you a bit of background in terms of ion mobility and where we're coming from. I'm now going to confuse matters by saying that, broadly speaking, there are about 4 different designs, and that's probably slightly inaccurate in that there are a number of hybrid designs in between these broad designs in terms of performing ion mobility. I'll start off with what I think conceptually is the easiest one to explain, and it involves a drift tube, as I've just explained, where we're going to accelerate our ions using an electric field and impede their progress with a drift gas going in the opposite direction. So this is the design that's favoured on the instrument that we use, which is an Agilent 6560 ion mobility QTOF. And you can have high pressure ion mobility tubes or low pressure ion mobility tubes. One of the big advantages of the low pressure tubes is that you can interface that more easily with mass spectrometry, which of course is taking part in a vacuum. The downside of interfacing the low pressure tubes with mass spec is that you're going to need a longer tube. You'll have less collisions as your ion goes through that drift tube. So you need to make it longer in terms of getting those number of collisions in there. One of the things to point out in terms of this design is where we find the drift tube on this design. So if you can see my arrow at the front end, on the left-hand side, you can see where our ion source is making the ions and introducing the ions into the mass spectrometer. And we've got our drift tube right at the front of the mass spectrometer. And then we've got a quadrupole here in this design, and then the TOF coming afterwards. So this is classically placed in front of the mass spectrometer in this particular design. The reason for saying that is that on one of the options taken by Waters on their Synapt mass spectrometers, they can actually place their design in between the quadrupole and the QTOF, and this allows them to do slightly different experiments that you do on the Agilent 6560 in terms of where this drift tube is placed. They've also used a different approach in terms of the ion mobility, and they use a traveling wave based approach. Because they have their drift tube in the middle of their mass spectrometer, they need to have a low pressure drift tube. And in order to keep that length down, they've got this novel electric field which creates a wave down the tube. So what happens is the electric field is applied only at small regions of the drift tube, and that application of the electric field goes down through the tube to create a series of waves which push the ions along. The analogy here is, if you remember back to going to the beach, if you stand on the beach when the waves are coming in, you'll gradually be pushed back into the shore. Now, as these waves of the electric field go down, they're gradually pushing the ions along as they move down through that tube. One of the advantages by placing the drift tube in between the quadrupole and the Q-TOF is that you can select some of the ions that are going into the drift tube and do that ahead of time of doing these experiments. Solution number 3 is differential mobility or field asymmetric ion mobility spectrometry. And this is an arrangement which is favoured by SciEx in terms of how they've been using ion mobility for doing lipidomics. And in some ways, this is, at least in my mind, is kind of similar to what would happen with a quadrupole in a triple quadrupole, in that you can use it to filter out ions, but in this case, in terms of their mobility, rather than their mass to charge ratio. So ions are transported through the device by a drift gas flow and then subjected to different field strengths in an orthogonal direction for different amounts of times. This means that ions are either deflected above or below the plane of the tube as they go through, and they get deflected in terms of their mobility. As a result, only ions with a certain mobility dependence can pass through, and this creates a filter, and it's very efficient in terms of the amount of space that it needs to use in terms of getting this filtering-based approach. The fourth solution I've got down here is trapped ion mobility spectrometry, and this is something that Sven spoke about yesterday in terms of his vendor talk, looking at TIMS TOF applications here. I sort of struggled whether this was going to be sort of next generation or current generation, because there's some quite amazing stuff that you can do with these types of TIMS spectrometry in terms of how you can move ions around in circles and past corners. The application here comes from Lucas Migas's website. So Lucas has been developing software tools for looking at ion mobility and also mass spectrometry imaging. And one of the applications that he shows alongside his software tools is this application for moldy TIMS TOF ion mobility spectrometry. So I thought this nicely goes with Zoltan's talk on mass spectrometry imaging. So in this case, again, we've got imaging here, but we're looking at two different isobaric ions here, and we use the TIMS part of the TIMS TOF to separate out these isobaric species and then create two different images. And then the surprise is that our red ions over here have a really very different distribution to what we might expect to see in terms of our green ions, in terms of the image that we have over here. And we, of course, can't separate that out by mass spectrometry, but we can do a nice job in terms of the ion mobility overall. So I've only got one slide of maths here, but I felt obliged to talk a little bit about the maths behind ion mobility, in that it tells you sort of why the CCS values are so useful, and also introduce resolving power in terms of ion mobility as well. So you can model how ions move down a drift tube very well, and we've understood the mathematics behind this, I think, sort of really since the 1950s, 1960s, and it all goes back to the Mason-Champ equation, which is shown over here on the top right-hand side. So we've got ion mobility, and ion mobility is dependent on the charge of our ion, as we might expect, and also this value inversely dependent on this value sigma, which is the ion neutral cross-section. So this is related to how big our ion is that's going through the drift tube and how it's hitting into our neutral gas ions as it goes through that tube, and that's where we get the collision cross-section from there sort of coming through. We can also define resolving power and relate this back to the properties of the drift tube length, the charge on the ion, to get an idea about what we can potentially sort of separate out in terms of these ions. And this is the equation on the bottom right-hand side, which is the resolving power of our ion mobility. So TD is the time in the drift tube, and then delta TD is the width at half height, the maximum width at half height of our peak as we go across there. To give you an idea about how resolving ion mobility tubes can be, so for a tube which is 15 centimeters long with 25 kilovolts across there, you get a resolving power of roughly 250 at 1 atmosphere of pressure. The downside, though, is if we're at 1 atmosphere of pressure, we're going to lose a lot of ions as we're going through there. And 25 kilovolts is a pretty large charge as well to put across those tubes. So typically we're operating at lower pressures, particularly if we want to interface with mass spectrometry, and we're trying to keep down the total length of our drift tube. So for the Agilent 6560, typically we're looking at a resolving power of around about 60 going on there, but I'll show you in my final couple of slides some much bigger numbers in terms of those sort of next generation ion mobility based approaches. So that's told you a bit about the physics, about how we can do this ion separation. Why would we want to do this for doing lipidomics? Because we've most of us have access to high resolution mass spectrometry, and we can do some chromatography on the front end. What can the ion mobility really add for us? So one of the big promises is that we can do isomer separation by ion mobility, give us a more detailed analysis of lipids. And I'd say that in terms of personal experience, this is certainly true, and I'll show you some examples of where this is true, but we've not been able to separate out some of the things that we really want to sort of separate out, I think, in terms of isomers. And I think that's going to wait until we get onto the next generation of ion mobility in terms of being able to separate out lipid species according to where double bonds are, and also begin to address whether we can actually separate out enantiomeric mixtures of samples as well. One of the things, though, that we can do right now with the ion mobility is that we can use the drift tube separation before fragmentation to increase specificity for our MS experiments. So effectively, you can clean up a sample in terms of the drift time and only sample across that drift time to get a better fragmentation pattern. We found that to be incredibly useful for assigning small amounts of phosphoethanolamines that are effectively sort of coeluting with phosphatidylcholines. We can separate them out, as I'll show you in a moment, in terms of drift time, and get a much cleaner MS spectrum, which is much easier to interpret. And I've pretty much said the final point here, but it's all about increasing confidence in terms of lipid identification. So from personal experience, I think sort of where we're at is that we haven't got the killer app in terms of some of those isomer separations, but it is helping us in terms of doing a better job in terms of lipid identification. Probably easy to sort of show you some examples of these. So this is, the examples I'm going to show you have all been performed by Christina Hines and Sonia Ligi, who are postdocs in my group. And if you want to have a good review of this area, they're also in a very nice review which came out of Erin Baker's lab, some similar examples, which is Kyle et al. From 2016. So I'd recommend that as reading if you're interested in mobility. So on the left-hand side, you can see sort of how we can separate out two different PEs over here that are isobaric in terms of their structures. Somebody kill their, could somebody kill their mic? Thanks. And on the right-hand side, you can see a nice job of where we're getting separation out of two PEs according to whether we've got a cis or trans double bond in this particular phosphatidylethanolamin over here. Slightly annoying that trans fats have now been removed from our diets, but we've got a really nice approach, I think, for sort of separating out cis and trans fats in terms of detecting this in phosphatidylethanolamin. I should point out that on the left-hand side, you could do this by fragmentation analysis, whereas I say that it's not always clear-cut from some of the data that you get from complex mixtures. Just like we were talking about yesterday, where Mikhail was talking about for retention times and how you can use chromatography to predict retention times for structurally related compounds, we can do the same thing for immobility. So this is a series of drift times plotted out for structurally related fatty acids. So on the left-hand side, we've got 3 different fatty acids that are based around C16, C18 or C20 chain lengths with variable numbers of double bonds. And it's not exactly a linear response across here, but you can begin to predict sort of how these fatty acids are going to behave in terms of their drift time or their CCS values. On the right-hand side, we've got a series of saturated fats where we're increasing the number of carbons as we go down through the fatty acids, and then seeing how that affects the drift time. And here you do see a nice linear response across most of these fatty acids. So just like we could do with retention times, we can use this as a sanity check in terms of what we're identifying against structures, and also predict where we might see particular lipid species. Here's an example of a series of phospholipids. On the left-hand side, we're looking at the effects of desaturation in terms of our fatty acid groups and how that affects the drift time. Probably more interesting on the right-hand side is we've got a series of different phosphatidylethanolamin here, and hopefully you can just make out in yellow we've got a cis phosphatidylethanolamin here and a trans phosphatidylethanolamin here, and you can see that you can begin to discriminate between the 2 of these species just down to whether you've got a cis or trans double bond that's present in the structures there. Phospholipids are great for separating out in phosphatidylethanolamin and phosphatidylcholines. Just to sort of show you here, this is our drift spectrum. Some people call them mobilograms as well. And we've got a phosphatidylcholine over here and a phosphatidylethanolamin. And you can see that we can nicely begin to sort of separate this out by mobility. I'll show you a nicer example in a moment of where that was really important. That comes out from the Kyle et al. Paper, so I won't go into this in any great detail. So this is the Kyle et al. Paper, which I heartily recommend, actually, if you wanted to know more about the use of drift tubes for ion mobility. Came out of Erin Baker's lab, and I think this is either figure 1 or figure 2, which sort of shows you nicely how complementary ion mobility is to doing regular for doing chromatography on the front end of your mass spectrometer. So in the top 2 plots over here, we've got normalized elution time across a series of species that we might detect. This is reverse phase chromatography here that we're detecting by LCMS. I think in this example, they've condensed down a 15-minute or a 45-minute run in terms of some of these lipid species down here. And then we've got mass to charge ratio over here. So the LCMS does a pretty good job of separating out different lipid classes, certainly the lysophospholipids, as we've already seen, and the triglycerides as well. But if you zoom in into the middle, you've got a lot of overlap between these diacyl phospholipids, which can make it very difficult in terms of interpretation. Now, if we come down to the bottom end of these plots, we can begin to look at ion mobility and how that varies across in terms of we've got mass to charge ratio on the y-axis and then ion mobility along the x-axis. And you can see sort of how we've got a nice trend across a range of different lipid species. And also note that it's a different type of separation than we're seeing with the chromatography and also with the mass to charge ratio to a degree. So we begin to sort of separate these species out and get improved confidence in terms of those identifications. Where I think it sort of comes into its own is dealing with things that are isobaric or things that are near isobaric, which we might struggle to separate out, even with high resolution mass spectrometry. So again, this is from the Kyle et al. Paper, and they've just zoomed in on a peak at 45 minutes in terms of one of their chromatographic runs. And they've put up here, I orientate myself the right way around, the mass spectrum versus the driftogram or the mobilogram along the x-axis over here. So if we look at the top part of this exploded plot, they're looking at two co-eluting lipids where we've just got a 0.036 Dalton difference. So you would probably detect that difference in, I think it's sort of 50 ppm or thereabouts. So you'd expect to see that in high resolution mass spectrometry. And indeed, you can see it as a little hump on the side of this much larger peak over here. But notice in the mobilogram, we do a much better job of separating out this phosphatidylethanolamine from this phosphatidylcholine. Below, we've got two co-eluting isomers. So it's the same mass, and we're not going to be able to detect that by high resolution mass spectrometry, at least just by straightforward MS. But what the eye-mobility allows you to realize is that you're actually dealing with two peaks over here by the fact that we end up with this broad mobilogram overall. Another nice example that came out of their paper is how you can separate out different lyso PCs. So here they're looking at lyso PCs, lyso PC 16.0 and lyso PC 16.1 in either human embryonic epithelial kidney cells or hepatoma cell line. And if I just orientate myself, this is in the kidney cell line over here. And when we're looking at the lyso PC where we've just got palmitate in there, we can begin to make out the difference between the palmitate in the SN1 position compared to the SN2 position. And similarly for the 16.1 in these lyso PCs for the epithelial kidney cell line. Where it gets interesting is in the hepatoma cell line, they again get two peaks for the lyso PC palmitate. But when they begin to look at the 16.1 species, they actually end up with four peaks. And they put this down to that they're getting differences dependent on where that double bond is present in the fatty acid in that lyso PC. So I think it sort of shows you the promise of being able to separate out structural isomers, but also cis and trans bonds in terms of these lipid species. This is an example from Ian Wilson. It's a metabolomics example, so apologies for not getting a lipidomics example here, but everything that I apply to metabolomics is certainly true for lipidomics in this case. So what Ian's been trying to do, working with Waters, is to try and produce a very fast, high throughput screening tool, in this case to analyze metabolites that find in urine, but you could apply this also to blood plasma based approaches. And this shows you what you get in terms of the extra bang for your buck in terms of doing the eye mobility. So what Ian has done here is put a 3-minute chromatography on the front end of his mass spectrometry, and he ends up with a lot of peaks which end up running together. So he's condensed down what would normally be a 15-20 minute run down to about 3 minutes. Now, without the eye mobility, he's able to detect just over 7,000 features, but bringing the eye mobility into that, he gets closer to 10,500 features. So he's beginning to get some of that separation back that he's losing in terms of his chromatography. And that's also then reflected in the pattern recognition models that he can build in terms of getting some of that information back and getting better clustering that's present there. So the final example is an example from my own lab, which is again work of Christina Hines and Sonia Ligi, and we collaborated with Valerie as well, sort of on this project, about how we've been using eye mobility for the detection of oxylipins and develop a workflow for oxylipins, both in terms of detection of known oxylipins, but also potentially flagging up unknown unknown oxylipins that might be found in our material. It's two case studies. The first one is salmonella infected murine bone marrow macrophages. And the second one is then human platelet activation. I won't go into the biology. I'm sort of more concerned sort of explaining sort of the workflow and sort of the advantages of what the ion mobility sort of gives you. At the time, we created an internal database for this analysis from 48 oxylipins that we had and fatty acid standards that we had sort of available to us. We acquired everything in triplicate and measured them under the same conditions that we're going to be measuring our eicosanoids in terms of mass spectrometry. Overall, we ended up with 275 species that we're detecting and roughly sort of 5 conformers per standard that we're detecting in terms of creating this database. Now, Sonia developed a NIME platform for doing this data analysis, and that was to allow us to bring in CCS values as a way of annotating some of the lipid species that we were going to detect. And in this NIME workflow, which you can find more details about in Metabolomics, which came out in 2018, and it's also available on Sonia's GitHub area. But the key to the workflow is that we can do matching of exact mass, matching of retention time, but also matching of CCS values for these eicosanoids. And if we get those 3 things matched, then we can be pretty certain in terms of our identifications overall. So this led to 3 levels of annotation. So the worst level of annotation for our basic approach is detecting something where we get an M/Z match with lipid maps. So here we've got an exact mass detection of an eicosanoid species, but we know that there could be a number of different structures that could fit that potential annotation. So that would be our lowest level of potentially sort of detecting something. In terms of identification, we were comparing to standards, and then we compared an exact mass, a retention time, but also the CCS value in terms of detecting that. And then we'd cross-validate that with fragmentation patterns as well. The other thing that the ion mobility gives you is that you can begin to flag up things as lipid-like flags. So we ended up with 2 categories where we had molecules which had an M/Z match with something in the library and a CCS match. So that would suggest that we're dealing with something that's structurally very similar to what we've already got in that database. Or alternatively, ended up with things with M/Z and retention time matches to things that were similar in the database, but very different CCS values. And that would tell us that structurally it was quite diverse compared to what we've analysed before. So this shows you the salmonella infected macrophages here. It's an N of 4, I think, sort of in terms of replication here, and the PCA analysis is rather overkill, so everything ends up sort of dotted on top of each other. But we can sort of nicely detect a range of different lipid species and identify them by matching the CCS values along with their retention times and also their exact masses, and confirm that with our fragmentation data that we'd also collected on the standards. Probably of more interest is when we look at platelets and platelet activation here. And the reason for that is that we can recreate the known biology that's present here. So we're looking at platelet activation on its own, and then also with aspirin treatment to look at different eicosanoids that are being produced. But we can also begin to detect some novel lipid species which are eicosanoid-like and aren't there in our library. So if I take you through the identification, first of all, of a particular thromboxane, thromboxane B2 over here, we can initially do a match in terms of its exact mass, which is shown in A. We can then look at the diagnostic product ions from this particular oxylipin, and then we can look at precursor and product ions, and we can use the drift time to align those nicely to make sure that we're not fragmenting anything else. The other information that we have is the CCS value, and we can make sure that all those things align. So it's a bit like climbing. we've got at least 3 points of contact at this point in terms of this molecule, and we can be pretty happy in terms of our identification of this particular thromboxane. However, at the same time, our ion mobility is also throwing up a number of unknown species. So we've labelled them 1, 2, and 3 here. So in this case, we've got an exact mass match for our particular within the library, different retention times, but very similar drift times. So this tells us overall structurally they must be similar, but the fact they have different retention times shows that we've got something that's structurally different in terms of the chromatography. So I think in the future this will be very useful for the identification of novel intermediates using these 3 or 4 bits of information that we have on our ions. Final example is something that we don't sort of do ourselves, but I've been incredibly sort of impressed by. But you can do ion mobility on intact proteins. So this is work that's come out of Carol Robinson's lab. And Carol was interested in how different lipid species might stabilize protein structures here. So she's looking at a native protein, getting it into the gas phase and measuring its ion mobility to work out how well folded this protein is. You can then dope in different lipid species here, and the one to sort of point you to is cardiolipins, which is CDL over in this panel C over here, gives you the most stability of these structures. And she was able to use this as an approach to show that cardiolipins bound to a particular protein of interest and increased its stability by measuring CCS values. I'm a bit surprised we don't sort of see more of this sort of in the field in terms of rationalizing why we see particular lipid species alongside particular protein species. So I think I'm on the home straight now in terms of ion mobility. I've got a single slide here on CCS databases. The CCS databases probably warranted slightly more slides, and there are a number of these. But the points I wanted to make is that you can now generate these collision cross-section values in silico, and they're very predictive for the ones that we measure in terms of our ion mobility. Indeed, you can get sort of accuracies of 1 or 2% in terms of these CCS values. So this is some work that's come out of Jiang Zhu's lab in Shanghai, all CCS down here, where there's a fantastic number of different species, lipids and small molecules, where they've predicted CCS values and then gone through and measured many of those to check that their values make sense. And there are other databases out there. So Bush Lab have a nice CCS database, as does the McLean research group. And now in terms of sort of the different vendor solutions for doing ion mobility, a lot of the vendors are producing software tools where they'll come with a CCS library that you can begin to sort of populate them with your favorite compounds as well. Just going to finish off on a couple of things in terms of next generations of ion mobility, and we've already heard a bit about this already earlier on in the talks. So this is from Waters, where they developed a cyclic ion mobility system. Around the cycle, it's 96 centimeters, I think, is the drift tube or drift circle in this case. But the fact that it's circular now means that we can do IMS to the power of n. So you can potentially sort of cycle your ions around there. And they're looking at sort of cycling ions around 100 cycles around this. And this gives you a resolving power of around about 750. So it's over a 10-fold improvement on many of the drift tube based approaches. One of the things, of course, you have to watch out for is that you begin to get wraparound effects. So some of your molecules will end up overtaking the other molecules as they go, some of your ions will end up overtaking other ions as they go around this cyclic tube. But we've got a quadrupole on the front end, so we can select for ions before they go into the circle. And it allows you to select ions, store them, and measure mobility in terms of that. Another approach, which again is sort of just being sort of commercialized, is SLIMS, which stands for Structure for Lossless Ion Manipulations. And this has come out of Pacific Northwestern National Laboratory from Dick Smith's lab. And here they're using a combination of DC potentials to trap ions on little circuits around printed boards over here. And again, you can get things to go around 90 degrees. They've got a couple of nice publications out where they're claiming resolving powers of around about 60. And I think that sort of recently just went up dramatically. I heard them speak and I couldn't find my notes on that. So I think, you know, that's one to watch for the future. So I've got my conclusion slide now. So hopefully what I've shown you is that ion mobility provides an orthogonal means of separation to mass spectrometry. And it's relatively straightforward in terms of interfacing ion mobility with mass spectrometry. I think the improvements in the approach will allow the separation of some isobaric species, and that's going to get better with time. So effectively, we're going through a period where we're going up at least by a factor of 10 for some of the ion mobility designs. So we're going to get much better separation. As I already said, ion mobility can be interfaced with mass spectrometry, and it can be interfaced at a variety of different stages in terms of high resolution mass spectrometry. We've also got CCS databases, and they can already predict a wide range of lipids. So hopefully that's encouraged you to give ion mobility a go along its various different forms. So I'm just going to thank the people that did the work that I presented from my own lab. So in particular, Christina Hines and Sonia Ligi, people at Agilent, John, Ruan, Sufyan and Will for all of their help for getting us up and running, and then Ian Wilson at Waters for the slides that he provided. And thank you very much.